These are the lecture notes for Chapter 11 on Mendelian patterns of inheritance. This is really our first chapter on genetics. And so the first section is a little bit of background information on Gregor Mendel, who is known as the father of heredity or the father of genetics. And then Gregor Mendel's laws that he developed through his experiments with garden pea plants. Section 3 focuses on Mendelian genetics that specifically applies to humans. And there'll be a section on human disease that is inherited. So, and then for section 4, genetics that we've learned beyond what Mendel discovered. So we'll start with talking about Gregor Mendel. You can't begin any genetics unit without learning about Gregor Mendel. And if you've had biology before, you've probably heard of him. Even if you haven't, you may have heard his name before. He's pretty important guy. Before Gregor Mendel's experiments, during the 1800s, people tended to believe in this theory called blending. And the blending concept basically said for any trait that the offspring would inherit a blend of the traits of both parents. But you can think of many, many examples where this just doesn't work out. I mean, I have a daughter who, if you've seen a picture of me, I'm a brunette, and that is my natural hair color is dark brown. I do have blue eyes, but I'm fairly short. I'm 5'4" and a little bit chunky. And my daughter is 5'10". She's tall, she's thin, she's very, very got very pale skin. Although I'm light skinned as well, but she's got very pale skin compared to me, and she's got blue eyes and blonde hair. She looks a lot more like her father. So she didn't inherit a blend, as far as the looks go. She didn't get a blend. So and you can think of many examples where there's a child that looks more like one parent than the other. Definitely not a blend of both parents. Or what if you have two parents who are very short in height and then they have a really, really tall child? There's just so many examples that don't work, that don't actually make sense with the blending concept. So Mendel wanted to discover exactly what was going on with inheritance. So his findings were in contrast with the blending theory. And what he discovered through his experiments with garden pea plants is he formulated the particulate theory of inheritance, and he also proposed the laws of segregation and independent assortment. So that's what we're going to go over in section 2. A little background on Gregor Mendel. He was an Austrian monk. So that one of his primary duties was to take care of the garden at the monastery. And he conducted his breeding experiments using the garden pea plant, which the scientific name for the garden pea is the Pisum sativum. And through those experiments, he formulated fundamental laws of heredity in the early 1860s. So you need to understand that he didn't know anything about DNA or proteins or chromosomes, or genes or alleles. A lot of these terms that we use so easily today, he just was able to understand how traits were inherited between two different garden pea plants that he would cross. So he would start with two different garden pea plants, look at the traits of each one, document the traits of the parent plant, and then cross them. And he would do that by using a paintbrush. He would take the pollen from one and apply it to the female part of the other plant. And if you aren't aware of this, plants have eggs and sperm. So the sperm comes from the pollen and the eggs are in the female part of the plant. So he would, he would cross the plants so that he knew which parent, which two parents were crossed with each other. So his experiments on the inheritance of simple traits in the garden pea disproved the blending hypothesis. Okay, so why did he use the garden pea? I mean, he was trying to learn about human inheritance, or the inheritance of traits in general among sexually reproducing organisms. But I mean, one would assume that he was trying to understand human inheritance. So why did he choose the garden pea? And if you think about if he was trying to study human inheritance, just how long that would take, he would need cooperation from individuals who had certain traits. He would have to get them to agree to mate, and it would take 9 months just to have one baby. So think about garden peas, which also have traits that we can study. They're easy to cultivate. In other words, they're easy to grow. They have a short generation time. It's only, I'm really not sure what that generation time is, but with humans, it's 9 months, and it's 9 months for one baby. Whereas the garden peas, it's just a few weeks or a month to, and you get 1,000 seeds that you can plant and grow within a short time and look at the traits of the offspring. So it doesn't take very long to get results, is what that is saying. Normally, garden peas are self-pollinating, but you can cross-pollinate them by hand, and this is what he did. He transferred pollen from the anther, or the male part, of one garden pea plant to the female part, called the stigma, of another plant. So he was able to control which plants he crossed and which traits he crossed, There were true breeding varieties of garden peas available. True breeding means they were pure for certain traits. For example, if you had a garden pea plant that was true breeding for purple flowers, it would only produce purple flowered garden pea plants. That means it didn't carry any other flower color that would show up in some of the offspring. So true breeding or pure varieties were available, so you could make sure that what you were crossing was pure for that particular trait. And the traits are simple objective traits. In other words, there's just two versions for each trait that he studied. Flower color, for example, you either had purple flowers or white. The color of the seeds, which is basically the garden peas that we eat, was either green or yellow. Believe it or not, yellow was the dominant color. We always use eat green garden peas, and we don't think that there could be yellow ones, but that's actually the dominant trait for garden peas is yellow peas. To show you how, okay, so this is just a part of a flower. The flower is the reproductive structure of a plant. So this is what a typical flower would look like with the petals removed, so that you can see the reproductive structures of the garden pea plant. So these 4 structures called stamens are the male reproductive structures. In the tips here, the tips are the anthers. That is where the pollen is produced. So you can take a paintbrush and rub it on the anthers and then transfer it to the tip of this structure. It's called the carpel. The carpel is the female structure. So when the pollen lands on the carpel, then it grows a pollen tube and it carries the sperm to these ovules, which are the eggs. And each one of the ovules, if they are fertilized by a sperm, they will become a seed. So this shows you how he did it. If he wanted to cross-pollinate one plant, he would cut the stamens off so that this plant cannot self-pollinate, like by a bee or, another pollinator transferring pollen from this plant to itself. So he would literally cut off the male structures from one plant. He would take pollen from another plant that he wanted to cross with this one, and he would brush it onto the stigma, which is the tip of the female structure called the carpel. And sperm from that pollen would fertilize these ovules, and he would grow a plant, or he would produce seeds. In this case, you can see this particular pea pod is green and the seeds are yellow. The pod itself can also be yellow and the seeds can be green. So those are some characteristics, as well as the petals can be purple or they can be white. But anyway, this is just showing you how he cross-pollinated. And he did this many, many, many times. And then once he got the seeds, he planted them so that he could look at the traits in the offspring. Okay, so in Mendel developing his laws of heredity, he used true breeding plants to start with. We call them the parent plants, or the P generation. The parental generation would be our original true breeding plants. He used varieties that only differed in one trait. So, for example, like I was telling you, he might cross a plant that had purple flowers with a plant that had white flowers, and everything else about them was alike. Or he might cross a plant that produced yellow garden peas with a plant that produced green garden peas, but everything else would be the same. So, and then down here it says, and by the way, this is called a monohybrid cross, when you are doing a cross and you're looking at only one trait. And the parents are different in just that one trait. So what it means here that he performed reciprocal crosses is that he would start with the parents, like, for example, a purple flower parent and a white flower parent, and he would cross them. Then the reciprocal part would be he would cross the offspring. Whatever the offspring were, he would cross them, and he would obtain a second generation. So the offspring are called the F1 generation, and then the offspring from the cross of the F1 generation are the F2. And from these experiments, he formulated the law of segregation, which I'll explain to you in a minute. But this is showing you his monohybrid cross that he did. So this is an example with the original parents being true breeding for, big T stands for tall plant. So that means this parent had two alleles, or two versions, the two versions of their gene that they carry for height are tall. So it's true breeding or pure for tall. And this parent carries two alleles for short. Okay? So we have a tall plant crossed with a short plant, and in the F1 generation, the offspring all of them are tall. But we know now that they inherited one tall allele from the tall parent and one short allele from the short parent. Mendel didn't know this, but he did know that all of the offspring were tall. And when he crossed the F1s, that means he crosses offspring that are tall, but they have come from a tall and a short parent. So what he ends up getting in the F2 generation is a ratio of 3-to-1. So about 75% of all the offspring are still tall, but 25% are short. So for every 4 offspring, 3 are tall and 1 is short. And that led him, and he did this with all these different traits. So tall and short, he ended up with about a 3-to-1 ratio of tall to short in the F2s. And for pod shape, you could have inflated or round pod or constricted pod. And he got a 3-to-1 ratio in the offspring. For every trait he studied, round and wrinkled seeds, yellow and green seeds, axial flowers, which are not on the tip, not forming on the tip of the stem or the branch, axial versus terminal flowers, 3-to-1, purple to white, 3-to-1, and then green to yellow pod color, 3-to-1. And then if you average all of those traits together, it was 2.98, which is close to 3-to-1 ratio. These numbers helped him to determine what must have happened. And so he went back and he was able to say, okay, so the two parents must have had